Great, thanks, Andrew. Can you hear me okay?
Yeah.
Can you see my full slide?
Yes, we can.
Wonderful. Well, thanks for the invitation to participate this morning. It's been a really informative seminar. I've really enjoyed it. And yeah, our topics have been all over the place, and this talk will be no exception. So I'm really going to talk today about the response to virus, both at a cellular level and at an organismal level, and how we believe here that the data suggests this contributes to COVID-19 disease. So lately I've been giving a few talks, and this opening slide of the global count of confirmed and people that have passed of SARS-CoV-2 it's actually quite a depressing slide to keep adding because these numbers climb dramatically. But I don't think I need to tell anybody here on the audience that this is obviously an important field of research that requires our attention. So today I'm going to talk to you really about mostly completely unpublished data that has been coming in almost on a daily basis here. Some of this data was literally processed yesterday. So please feel free to ask questions, and I will apologize in advance should I not have thought of something ahead of time. But I'll break my talk down into really, hang on here, oh dear. All right, so this is the virus we're all talking about. I'm just gonna skip ahead of this. This was what was identified as SARS-CoV-2. Looks a lot like SARS-CoV-1. The way it works is it comes in basically as a contagious mRNA. It first drives this first open reading frame called ORF1a, which gets processed into a lot of smaller proteases and pieces for the replication machinery. It also has this second open reading frame attached to it, which demands that the ribosome shift right here, which doesn't happen very often. So you get a lot less of the ORF1b proteins, and this is where the RNA-dependent RNA polymerase is, and a number of different replication machinery. As the infection proceeds and you have enough replication machinery to advance further, you then start making an anti-subgenomic material. So the polymerase obviously starts making 5' to 3' on this side, and does this pretty amazing trick where it starts replicating the RNA, and it comes to a transcriptional regulatory sequence, or a TRS, and it literally jumps off its template and then reprimes on the other side. And so these are all these subgenomic RNAs that produce the famous spike, which is of course on the outside, the envelope, the matrix, the nucleoprotein, and then all these other accessory proteins which just have the term open reading frame with a number. And these are, we'll discuss them in a bit, but a lot of them are immunomodulatory genes that the virus uses to control how the host responds. This is like a very high level bird's eye view of how this whole process proceeds. You bind ACE2, you get endocytosed. The ribosome sees the RNA as messenger RNA and begins transcribing and begins translating ORF1a and ORF1b. Until you get to that later phase where now you start making these subgenomic RNAs. And this is all pertinent for today's talk because I'm really going to tell you today about how the cell knows it's infected by this particular virus. And I think a lot of the source of that detection comes when you start having this polymerase jumping template and generating a lot of RNA that is highly complementary to other pieces of RNA. And of course, this is essential for virus because it needs to make all of these accessory proteins, but this is really the weak spot with regards to how the cell might recognize that virus is there. And it does that by these pathways. So this is something I've been studying for 20 years. But in general, again, this weak spot right here where you're producing your accessory RNAs, you're generating a lot of so-called PAMP, or pathogen-associated molecular patterns. And the cell comes equipped with two intracellular sensors. They are RIG-I and MDA5. Long story short is RIG-I is thought to recognize RNA with triphosphate ends, and MDA5 is thought to recognize longer double-stranded RNA structures. But in either case, these oligomerize on top of that substrate that they see, and the oligomerization ends up allowing them to bind to this other antiviral protein called MABs, which sits on the mitochondria, and it assembles this massive complex of kinases, including the IKK alpha and beta kinase complex, which is important for NF-kappa B, which is depicted here, as well as the IKK-related kinases, which is TBK1 and IKK epsilon, and they activate IRF3, but they can also activate IRF7 once you have interferon signaling in place. And so these two major transcription factors, the IRF3 and NF-kappa B members, they come and they form something called the enhanceosome, and they drive the production of type 1 and type 3 interferon. So this is really the beginning of the antiviral state. And so interferon comes out, it gets released from the cell, and it can signal to the neighboring cells, it can signal to the same cell, and you get this signal transduction event, which culminates in the activation of a second set of transcription factors. We call this one the interferon-stimulated gene factor 3, or ISGF3. And it's composed of STAT1 and STAT2, which is associated always with this IRF member called IRF9, which dictates its DNA binding specificity. And so these guys go into the nucleus and they induce anywhere from 200 to 300 genes. And we generally refer to these as interferon-stimulated genes, although it is important to note that the IRF3 and other members of the IRFs can also induce many of these responses. And that's because the IRF family is essentially assigned this family member by 5 tryptophan repeats, which dictate its DNA binding domain. And so what IRF3 binds is very similar to what this complex binds because it has an IRF in it, for example. But in any case, this entire cellular response really has these 2 major arms. So the interferon aspect of this transcriptional response is really the call to arms. It is the telling of the neighboring cells that there's an imminent threat coming, and it includes the induction of many direct viral effectors. So this includes things that are going to block translation, cut up double-stranded RNA, maybe prime cells for apoptosis. They include inhibitors at the membrane to stop viral egress or to encapsulate the RNA-dependent RNA polymerase. The second aspect of these infected cells that require a response is something I hear called the call for reinforcements. And this is a barrage of cytokines and more noteworthy chemokines that are really there to create a trail for the more sophisticated arm of the immune response, that is the adaptive immune response, to come to the site of infections. And so this starts off with things like neutrophils, but then ends up being more like macrophages, B cells, T cells, and this is all a work towards gaining a specific response against the pathogen that induced it. All right, so initially we started this quite some time ago, and we first threw this virus onto A549 cells. This is a human lung carcinoma cell. We generally like this cell when we study respiratory viruses because it has an intact antiviral response, which is not true for many cell lines that we propagate. But A549 has essentially no ACE2, which is the receptor for this virus. And so if you throw SARS-CoV-2 onto A549 cells, we see very limited entry and very limited replication, as depicted here by this essentially a histogram of RNA. It does appear that you can get some entry, and it's unclear whether or not this is just through random endocytosis that occurs, or whether or not there is some low level of ACE2 on a subset of cells. But certainly when you just take A549 and you add ACE2 to these cells, you go by 3 orders of magnitude increase in the amount of virus you have. So this is all poly-A-based RNA sequencing. So this is, you're looking at essentially an mRNA, and you're looking at the number of reads across the viral genome. So you can appreciate here that really we see in general this kind of dip where the higher peaks are coming from here, which probably is in part due to the RT reaction, in part due to the fact that you do make a lot of the accessory proteins. The other point I wanted to make here is that you might notice that the virus plateaus here. And I think if I could have a single take-home message, this is going to be it, that when you come in at an MOI of 0.2 and wait 24 hours, or you come in at an MOI of 2, essentially you produce the same amount of virus, which is a scary amount of virus. We see that the virus can occupy as much as 40% of all polyadenylated RNA in a cell. Again, I want to bring to your attention that this amount seems to max out at about 40%, regardless of whether you started a low MOI or a high MOI, and this will become relevant in 1 minute. We can also use that sequencing data and look for those gene junctions. So as the jump I mentioned of the RDRP, you can map those specific reads that have those, and therefore you can follow all the accessory proteins and see that in fact they are all being made in response to either an increasing MOI or a kinetic experiment. So I think my new favorite piece of data is this one. I'll spend a few minutes on it. But this is just simply RNA-seq data of A549 cells expressing ACE2. This is, they replicate robustly. They cause CPE. These cells will all die. But there's an amazing dichotomy here between the postdoc who did this work, Ben Nielsen, did an MOI of 0.5. So this is one virus for every 2 cells versus an MOI of 2, which is 2 infectious particles coming in per cell, at least mathematically speaking. You can see down below that, again, very much in agreement with the form I just showed you, that an MOI of 0.5 and an MOI of 1 generates about the same amount of SARS-CoV-N. So it is essentially reaching the capacity of using all the resources that the cell is offering it. But one of the really big differences between this is that while you have a strong transcriptional response to virus in both cases, in MOI of 0.5, you see cytokines, but no evidence of interferon. So there are a few scattered ISGs in here, which could be induced by interferon or by the activation of IRF members. But you do not see interferon beta, which is a type 1 interferon, or any of the interferon lambdas, which there are 3, in any of this data. However, the exact same experiment done at the same time, but just increasing the MOI, you see that interferon beta is actually like the 7th most regulated gene, and you can pick up all the lambdas in addition to the cytokines. And so the difference between seeing interferon in your data set or not seeing it really reflects the incoming MOI of the virus. And I'll explain to you why I think that is. This data can also be generated by, of course, non-transformed cancer cells. So these are lung organoids, which is a nice model to look at replication. They're depicted down here, so they express ACE2, and here you can see staining for SARS spike protein. And again, you can appreciate that we have full coverage of virus. We have a higher peak at the end, demonstrating the accessory proteins. And when we perform RNA-seq on these lung organoids, we see, again, evidence of a lot of chemokines. So we see that call for reinforcements, but we really don't see the call to arms. We don't see a strong ISG signature or, of course, any evidence of type 1 or type 3 interferons. So if you speak in terms of low versus high MOIs, you could just simply say that in an organoid system, primary human cells, the infection mimics much more of a low MOI condition. All right, so why does MOI matter? Well, that's simply because, like every major human pathogen, in order for this virus to survive in nature and be successful in evolutionarily speaking, it has to be able to deal with this antiviral response that we've created over evolutionary space. And so, while I'm sure there are many examples of this for SARS-CoV-2, we only have 2 well-published examples at this time. One of them is, in fact, this protein called ORF1a, which is a tiny protein. It's only 20 amino acids long. This was posted on bioRxiv recently from Cato's group in the University of Osaka. And really all he wants to show here, so this is flu, and this is flu's antagonist. And if you express flu's antagonist, which is a double-stranded RNA binding protein, you can see that if you throw in a virus which is relatively clumsy with regards to its capacity to antagonize the host antiviral response. So this is a centivirus, so it's a virus that's evolved to do very well in mouse. It's a great inducer of interferon, but you can see with the addition of NS1, you can completely knock out the induction of, in this case, an interferon beta promoter system. And similarly, if you take ORF1b from SARS-CoV-2, you can see that in a concentration-dependent manner, the more more ORF1b you accumulate in your cell, the more potent you are at shutting off these defenses. And so most of you are probably already going to figure out where I'm going with this. And that is, long story short, is if you come in with a low MOI, you're not going to generate the PAMP that's responsible for the induction. So it's a timing issue, because basically you need time to make things like ORF1b so that when you as a virus that's replicating start inducing triggers for the induction of interferon beta, you have the capacity to shut that down. And so what is the trigger for SARS-CoV-2? Well, like SARS-CoV-1, it seems to be the production of double-stranded RNA. So this was again done by a postdoc in my group. Same one, actually, this is Ben Nielsen's work, but this is simply showing that the green signal here is this J2 antibody. It recognizes double-stranded RNA specifically. And you can appreciate that in the presence of virus infection, you really light up the cells with this double-stranded RNA. So again, this probably relates to the fact that the subgenomic RNAs require that jumping mechanism, and that inherently generates double-stranded RNA, which should induce the interferon response. And so again, this goes back to this low MOI, high MOI conundrum, that at a low MOI, we do see mostly a cytokine signature, which is largely an NF-kappa B driven system with a spattering of interferon stimulated genes, suggesting that most of the interferon response is shut down. Whereas if you come in the same cells, same infection, you now see, you know, the who's who of the interferon response. And so there really is a dichotomy between these two. These are slightly different cells. If we knock out the two major detectors, so for example, if you knock out RIG-I, it really makes no difference. We still see all of the major players, both in the chemokine space and in the interferon stimulated gene space. But if you knock out MDA5, again, the sensor that recognizes double-stranded RNA, we essentially lose all this response, and all we're left with is a spattering of cytokines. And so it does seem quite clear that the way this virus should be recognized is MDA5, and the difference between whether or not you have an interferon signature or not really depends on the timing of replication and your capacity to make those accessory proteins. And so just to go back to this model, this is this is one other example. This is a protease that's thought to block the activation of IRF3. And then we have this should be ORF3b, which blocks the some aspect of this signaling pathway. And then all the other proteins in red are all other antagonists that have been shown for SARS-CoV-1 by Peter Palese here at Mount Sinai, by Matt Freeman, by Ralph Barrick, by many others, to really interfere with different aspects, both in this space and the induction of interferon, but as well as the response to interferon. And this would also shut off interferon stimulated genes. For example, ORF6 has been shown to prevent the shuttling of this factor, so you don't have the capacity to induce interferon stimulated genes, even if you do successfully launch some interferon. All right, and so we've been working quite a bit trying to understand, you know, beyond just simple cell model systems of how this really pans out with regards to animals.
2 minutes, Ben.
Oh boy, okay, so we started with ferrets. I'm just gonna blow by ferrets. This is already published anyways. Basically, ferrets are an interesting model. We don't see interferon, we see a lot of cytokines. And we don't really see a lot of tissue damage. So to me, it's not the best model. I like hamsters a lot better. So these are hamsters, the golden Syrian hamster. And in lung and in trachea, the virus replicates really well. We see a little bit of olfactory bulb replication, which may explain why we get the smell phenotype that we see. It's not that hamsters lose weight, they basically stop gaining weight. So there is some evidence here of morbidity, that they don't feel very well. They're probably not drinking water. And hamsters, actually very much like an infection, they look a little different. So their interferon stimulated gene signature is a little stronger, but we still don't see type 1 or type 3 interferon like you would imagine. And so my guess would be that this infection initially probably is higher than it would be under physiological conditions, because we're administering 10 to the 5 PFU per ml. But this infection clearly is cleared by the hamsters, just like it is for ferrets, and results in this leftover cytokine signature, very reminiscent of what we see in, say, COVID-19 patients. And so, again, this is published, but if you look at the lungs of COVID patients, or you look in the serum of COVID patients, it's quite clear that you have a very strong call for reinforcements. We have very strong activation of NF-kappa B, but the IRF arm of the defenses is clearly handicapped by things like ORF3b. And this ultimately prevents you from controlling the virus at the site of infection and leaves just nothing but infiltration, which is what we see in COVID-19 patients, with massive infiltration of both neutrophils and macrophages, and then of course all the lung damage that ensues after that. And so this is just looking at cytokine levels in the serum of COVID-19 patients. The red are people that tested positive for COVID-19. In black, they are people with respiratory infection that tested negative. And you can see here that you also have a very high signature of all the same chemokines that we see in ferrets, that we see in hamsters. There is definitely IL-6 seems to be an important marker for both ferrets, hamsters, and in COVID-19 patients. So it's a very strongly induced. My major conclusion here is that at a low MOI, we see that the virus essentially wins. It has the capacity to block MDA5 mediated interferon response. The infection likely triggers NF-kappa B in different ways, which explains the high cytokine levels. And based on the hamster and ferret models, which really are modeling younger, let's say, teenagers getting SARS-CoV-2, the fact that they both recover well and the signatures that they're inducing would suggest to me that SARS-CoV-2 and the weak response that does ensue, so the few ISGs that we do see, must be sufficient to control local infection to enable your adaptive immune system to neutralize the virus. Whereas perhaps in individuals with comorbidities, this same initial response is even weaker, and it just completely fails to control local infection, which ultimately just exasperates the immune response as the more professional immune cells start coming into the lung. And so with that, I really would be remiss without thanking the people that do all this work. None of them are me. So Ben Nielsen, Daisy does all of our hamster work with Rasmus, who is pictured over here. This is Daniel Blanco. He does all of our bioinformatics with David Sachs, who's not pictured. This is Ben Nielsen, who does a lot of the PRR work. We have two groups from Cornell, Rob Schwartz and Xubing Chen, who have done helped us with all the organoid work, and of course, our sponsors. And I am sorry that that came off to such a rough start, but hopefully I came across with a few interesting points, and I'm happy to take some questions. So thank you.
Thank you, Ben, for a very interesting talk. And we do have time for some questions. I wonder if you could comment on the mechanism of the highly prevalent asymptomatic individuals.
Sorry, you want me to comment on the highly prevalent
asymptomatic? Why, you know, what is the cause of the asymptomatic type of infection? Why are some people asymptomatic yet, you know, high shedders of the virus, and what is the molecular basis for that?
Yeah, it's a good question. I don't know that I have data to support it, an answer to that question in a good way. So with ferrets and hamsters, if we base a lot of information on that, you know, you do have to be a little careful in that it's unclear whether or not the antagonistic strategies of the virus are as efficient as they would be in human cells, for example. But if we do suggest that that data is an accurate depiction of what happens in younger individuals, basically what you're looking at is a very suboptimal interferon response. And so if you don't control virus infection, so my view of the way this works is initially you require those interferon stimulated genes, and they're not really there to clear the virus. They're really there to slow the infection at the site of replication, to buy you time for the whatever 5, 6, 7 days it's going to take for you to bring in antigen presenting cells and start developing specific captured responses to epitopes. And so if you are generating an inadequate response, you're not controlling spread. And so I think the virus just continues to percolate. And so maybe the difference between high spreaders and low spreaders are simply whether or not where you fall on that spectrum of what is your limited induction of the interferon response in any way adequate to control infection or not. And you know, I think you're right on the cusp of being able to control it. So in ferrets and hamsters, you know, we don't see high levels of interferon, but we do see a good spattering of ISGs, which clearly is enough for them to control it, because virus peaks after 3-4 days, and then it goes down and it's cleared, which is what we see in teenagers, for example. But if, as you get older, those responses dissipate, that basic innate mechanisms dissipate, then you don't control virus. And so even for things like super spreaders, you could imagine that if you don't control that because there's some genetic predisposition that lowered that particular response, you would maintain a lot of virus that would allow you to shed it. And then I guess being asymptomatic is a tougher one, because the symptom aspect of virus infection really comes from the second arm, the call for reinforcements. And so maybe in an absence of a strong interferon response, your virus is navigating through your respiratory tract so that maybe it stays ahead of the adaptive immune response, but it's all speculation. I can't really speak to that other than
Ira, Ira Tabus, if you
Yeah,
unmute.
Ben, I just wanted to clarify something. So you made the beautiful point that at low MOI, that dampens the interferon response. So can you just review again what happens at high MOI, particularly with regard to the fact that that is likely going to cause worse disease?
Right. So so at a high MOI, there's two dynamics, especially in cell culture, that happen. So one, so MOIs are measured by plaque assay in viro cells, right? So that's infectious particles. And so of course, you're only measuring infectious particles, and there's always also a population of non-infectious particles within that mix. So some people refer to them as defective viral genomes. They're usually just broken pieces of virus that are partial, and those can be really active inducers of the interferon response. And so one difference between a low MOI and a high MOI infection is you're also introducing a lot more defective interfering particles, and you're introducing those immediately. So right upon infection, those infectious particles can trigger MDA5 before you had, as a virus, had any chance to actually stop the signaling that results from those DIs. Even in the absence of those defective viral genomes, though, coming in an MOI of 2, because the biology of the virus is first to make replicase, the virus prioritizes in many ways. First, it makes ORF1a. It purposely requires a ribosomal frame shift to make the RDRP, so it's trying to balance how many replicase machinery products it makes, and then it wants to make accessory proteins in that order. But if you come in at an MOI of 2, the way viruses balance between early and late infection is usually based on the amount of a given protein that you have. And so if you come in with 2 infectious particles and maybe a broken DVG that can make some replicase, you throw off the very biology of how that works. And so again, you're going to start making double-stranded RNA before you have a chance to make an antagonist. And so while that's really easy to model in cell culture, that same dynamic is going to happen in vivo as well, that in your initial infection, you have maybe a few infectious particles that catch in your upper respiratory tract. And as the virus replicates, initially it's probably unlikely to induce any interferon, but of course, as infection progresses further and further on, your local multiplicity of infections skyrocket as you release 100,000 infectious particles at a site of infection. So the dynamics are constantly going back and forth. And I think it really has to do with how the speed at which the virus replicates, spreads, the burst rate of that virus, and how much time the virus has to antagonize your response.